Hello, folks. I'm going to be recording this lesson, which is about experimental design. You all have some guided notes to work through as I'm presenting this. A lot of this shouldn't be new at this point in your science career. You've probably at some point talked about independent variable, dependent variables, hypothesis, things like that. But the guided notes you'll find will be in the module about experimental design. That's under the experimental design lesson. There are guided notes there, so you can click on those and you'll just be basically filling in the blanks. Yours will look a little different than mine, but yours should look something like this. So there will be blanks that you'll be filling in, typing as I'm presenting the material. Okay. So this lesson is on experimental design, which is just a part of the scientific method. It focuses mostly on the procedure and the analysis of the data and so on, and writing a good conclusion. So those are our focuses this year. And in this experimental design, it works its way. It's just a step-by-step approach to go through and solve a problem. And then as a further assignment, you will be performing these steps and designing an experiment at home. Okay, and writing up a report about it. So in an experimental design, this is just a step-by-step approach. I'm going to come up with a hypothetical situation, a lesson, or an investigation, and we'll work through these steps as we try to reason through that. All right, so the first step in designing an experiment is to know what you're trying to solve. What's the purpose? What's the problem? So what do you want to find out? You want to write a statement that describes what you want to do. For example, let's say I'm curious about how well plants grow when you add different kinds of soda to them. All right, so let's add Coca-Cola. So the caramelized stuff. Let's add Sprite. Okay, to one plant. So Coke to one plant, Sprite to one plant, Mountain Dew. We'll add that to another plant. Okay, so we'll have these plants. We want to have these plants, and we want to add. These are my plants. See how I'm an artist. And we're going to add these different types of drinks, different types of carbonated beverages to them. Okay, so we've got a yellow one, we've got a white one, we've got a brown one. Let's get one that's kind of in between, so like L8. If you don't know what that is, that's a drink that's bottled in Winchester in Clark County. It's a Kentucky made and Kentucky owned bread type of drink, and I grew up on it, and it's good stuff. It kind of an acquired taste. It's a little bit of a ginger ale, so that's the name, the Ale8. But it's a wonderful drink. All right, so we've got our problem. I want to see if any of these help plants grow. Now, one of the things that we'll think about as we look at these is how they're different and how they're similar. They're all carbonated. Okay, so that's a constant. They are all liquid. They all are water-based. They are all full of sugar. Okay, the main difference is we've got caffeine in 3 of them and no caffeine in another. Okay, difference in color, difference in there are other differences, but really the caffeine is a change too. So it might be best, if we're going to maintain the integrity of this experiment, is to try to find a clear caffeinated beverage. So I can't really think of one right now, but I'm sure there is one. So we'll get rid of Sprite for now and keep these 4. Okay, so we've got a dark one, we've got a yellow one, we've got a sort of a tan one. And as I work through this, maybe I'll think of another that we can add to the list. But to just try to keep it as consistent as possible, okay, one of the things that we're going to define down here later on is that we want constants. Okay, we want things that, and we want to show those constants, and we want to reason through these different types of beverages. Okay, so we've got a problem. We want to know something. We want to learn something. Okay, that's the whole point of the scientific method, is for us to, in experimental design, is to learn something. The next step is, once we've learned it, we need to define some variables. And I've been playing with that as I've talked about this little hypothetical anyway, as I came up with the problem. Now, one of the things that you'll want to do before you start defining variables is maybe do some research. Okay, so it might be that somebody has already done this particular experiment. They've already tested plant growth with different types of soda, carbonated beverages. Okay, so you might want to do some research. You Google it. You try to read scientific papers. You hunt for any evidence that this has already been done. Let's assume I've not done that. So let's assume that it hasn't been already studied. Okay, so we're moving on then to the defining the variables. So in the scientific method, we've come up with this question. We've done the research. We've established that it isn't something that's already known, okay, that's already been studied. And now we're coming up with starting to build our experiment here. As we build our experiment during the research, we're going to end up with a hypothesis. And that's coming up in step 3 there. Okay, so we're going to define our variables. The first variable that we need to understand is what we are actually changing. Okay, so it was this list of, in our case, it was this list of sodas. All right, so the independent variable is the variable that we are in control of, that we are manipulating. It's the one that you will have different adjustments that you're making to it. And then any measurements that you take, whatever you're trying to solve this problem, any measurements that you take that depend upon that is your dependent variable. So what's going to depend upon these 3 or 4 different types of soda that we've got here? Okay, what depends on them is plant growth. Okay, so that's going to be our independent or a dependent variable. Our independent variable was the different types of soda. Our independent or a dependent variable then and remember, we pick these based on what we're trying to solve. We don't want to pick a dependent variable about like how well they taste, okay, or how much they'll keep you up at night, or there's other studies that we can do with these 3 different types of soda, but what we want to focus on is we want to solve the problem. The problem is, for our purposes, which will make the plants grow faster or better or higher, okay, but we got to define that too, how we're going to measure that. So when it comes to the helping the plants grow, what do we want to help them? How do we want to measure that? Or we want to measure that with height? Okay, so in that case, we would have actual data that we could measure with the meter stick or a ruler. Okay, so height could be a dependent variable. Speed, okay, how fast they grow per day. So rate of growth, which it has this height dependence, but it's also going to have a rate to it. Lushness, okay, how lush it is, how good it looks. Okay, so if we're going to try to do this, how to make a plant grow better, we need some way to measure that, and that's got to be agreed upon by your research, okay, or in your investigation. You've got to understand how you're going to document that one or more grow better, okay, or none. So the dependent variable in our case is going to be, let's say, height. So it's going to grow faster, basically. All right, so we've got our independent variable. We've got 3 different types of soda and potentially 4 or 5 or however many we can think of, all need to be caffeinated. And the only difference that we can have between them, and there's probably differences in sugar content, but that may be going deeper than we want to, trying to figure out how much sugar there is per drink. Okay, so really right now, the only difference we have between these, of course, is flavor, and those are compounds and chemicals that could influence it, and color. So right now, we've maintained it as best we can by taking out that caffeine part, because what we've got going against us, if we're setting up an experiment, we only want to test one variable. Okay, that's the independent variable. We only want to test that, and we only want to have one dependent variable that we're focused on. We only have one way that we're measuring that. What we don't want is for other factors to come into play here. Okay, and those are called our constants or constant variables. So these are all the factors in the experiment that we don't want to change, we don't want it to allow it to change. Controlling these constants is part of our procedure, okay, is how we're going to maintain that. So when we set up our procedure, we want to make sure that in there, we talk about that we want to use, of course, think about some constants with me, some things that we don't want to change. In our case, we don't want the plants to change. We want to use the same plant, same type of plant, a fern or some kind of flowering plant, a tomato plant. We want to use something that's similar, okay, same type of plant, basically the same height to begin with, planted in the same soil, with the same sunlight. So everything that we're trying to maintain the same with all of these, so that we don't want those to become a factor, is our constants. Okay, and we've got to think through that. That's a brainstorming session, that you want to come up with as many constants as you can when you do an investigation, so that even a simple investigation like this, so that you try to maintain all those so that they don't contribute to your results. The only things that we want to affect our results is the type of soda. Okay, that's it. We don't want anything else affecting it. Now, this is sort of a biology type hypothetical kind of thought investigation or experiment. And in a biological type of setting, you're often going to have a control, okay, or a control group. Now, a control or a control group, and it would be a group if you're doing like a testing a vaccine. We're hearing a lot about that right now, aren't we? Or you're testing the effects of a drug, okay? There are the side effects of a drug, the effectiveness of it. We would have a control group. And usually in the case of some kind of pharmaceutical situation, it would be a double blind type of system. But we would have a control. We would have something that we're using as a placebo. So a good control here with these would be in a control. You treat it normally. You treat it with something that you're going to see how it changes. Okay. Now I would suggest, of course, since we're working with water or working with plant, that we try water. All right. But water doesn't have some of these other factors. Okay. So we might then try some kind of just seltzer. All right. So it's got the carbonation, and it might be, we might be that we're just testing to see if the caffeine is what affects it. I think water is probably the best. So let's use water. Water, of course, would be as the normal thing that you do a plant. Maybe none of these actually improve the plant's growth. Maybe they actually hinder the plant's growth. And we would only notice that if we were treating it with something normal. Okay. So in that case, our control group would be the plant that has water in it. So we've got now 4 liquids that we're testing, but we're only testing 3. The other one is the control. So the other 3, the the Coke, the Ale-8, and the Mountain Dew would be our experimental group. So the ones that we're actually testing the dependence of. Okay, so with the experimental group, it's being tested with the independent variable, and they're only one factor different from each other. There could be more different, of course, because we got the flavor compounds, but we've narrowed it down as best we can. Okay, we've looked at that as best we can. And another thing that goes along with this is, and these aren't really step 2, defining the variables. These are just important parts in the experiment. That's part of it. What the control group is going to be. The experimental group, of course, is what the independent variable is all about. Okay, and then we have repeated trials because we want to make sure that we're not having a fluke. Okay, so we're not just going to have 4 plants. We may have 12 plants and 4 sets of 3 that so that we can get sort of an average, okay, within a particular group. Otherwise, it might be a fluky plant, and this plant just loves to grow, whatever we're giving it. Okay, so we want to make sure that we use some repeated trials. The number of times the experiment is repeated. You never want to just trust one trial to it, one outcome. Okay, you want to make sure that it's repeatable, that it happens, and theoretically, you might try with these independent variables, different types of plants as a kind of a follow-up experiment. We wouldn't do that right away. We'd pick like a marigold or something, and we test this over a couple of week period with 12 different marigolds. And then once we've analyzed those results and seen what that happens, we might try it with a different plant to see if it's just the marigolds like Ale-8. I don't know. I'm just guessing here. All right. So once we've established all this, we do want to come up with a hypothesis, and this is important. Okay. It seems like it's not a crucial step in this. If we're really going to test them, we're just going to see what happens. All right. But we do want to take a stand. We do want to assume or predict that one thing's going to happen, or that of a particular outcome. It's an inferring statement that can be tested. Okay, so we do want to make this decision. And since we don't know, unless you all have experience with growing plants with different types of caffeinated drinks, you might have a leg up on me, but I don't know what's going to happen. So I'm going to predict, because I grew up on Ale-8, and I think that it's one of the best drinks out there. I'm going to make a prediction that if we supply a marigold plant, or a set of them, 3 marigold plants, with equal amounts of these different drinks, okay, in different sets of these marigolds, and all of they're getting that's different is the different drink, okay, not the amount, not the temperature, okay, nothing like that. We want to maintain all those as constants. Then Ale-8 is going to be the best. Ale-8 will make the plant grow higher. Do I know for a fact that's going to happen? No, I'm just predicting. Okay, it's a prediction step. Often, these could be in an if-then type statement. If we do this, then this will happen, but it doesn't have to. Okay, there's no rule for that. It can be just a blanket prediction. Now, our goal here is to see if this prediction is correct. All right, so we're going to set up our experiment, set up our procedure in such a way that we can test this hypothesis. Will Ale-8 be the best drink to give to the marigold plants? Now we want to design the experimental procedure. All right. Now, you all have had experience, I'm sure, with hypotheses and coming up with an independent, dependent variable. We'll talk later about graphing those. And you've probably had some experience with a procedure. But I want to make sure that with our procedures this year, okay, our focus this, or at least the early part of this year, is to write the best procedures we can. Now, in chemistry, if we were at school, we would be doing investigations in labs. Labs tend to have a procedure given already, okay? But a true investigation, of course, you would be writing your own procedure and coming up with your own plan. So with this plan, with this procedure, you want to make sure it's very, very, very detailed. Okay, so we want to make sure that it includes lots of details. We want to overkill the amount of information that we're giving here. Okay, we want to select only one thing that changes. That's our independent variable. We want to change that variable that will change a variable that will help test the hypothesis, and that's we're going to do the height and the different drinks. The procedure must tell how the variable will be changed. Okay, so the different drinks here. What are we doing? We're providing this. We want to be specific about how much we're adding every day. If we're doing it every day or every other day or twice a day, we want to tell all that. We want to tell how we're measuring it from the soil to the tip of the last leaf, okay, or we're measuring it side to side. We want to collect that data, what units that we're using, okay, all that needs to be in the procedure. So it needs to how it will be measured. It needs to indicate how many trials you're going to have. It must be written in a way that somebody can actually follow it, okay, like a recipe. Recipes don't just say, take all the dry ingredients, mix them together, throw the liquids in, mix those together, throw it in the oven, and bake it till it smells good. Okay, the recipe gives you details about how all that happens, which ingredients you add, when, how long you stir them sometimes, how high you turn the oven on, what temperature you use, how long you cook it, what kind of pan you use. There's lots of details in a recipe if you're going to cook something. All right, so there needs to be those kind of details in a procedure. On top of that, and this bullet isn't here, so we're going to add it. On top of that, then you may need to jot this down. I didn't think about that. I'll look at the guided notes and make sure that they parallel this. Okay. On top of that, you need to make sure that you show how you're maintaining the constants. Those are not allowed to change, remember. So all that brainstorming that you're doing, trying to make sure that the only changes that we're making are to the type of drink, okay? Everything else is the same. We want to tell about that in our procedure. We don't want to just assume that the person carrying out this experiment is using the same type of soil, okay, for all of the plants. We want to, if we're feeding it with a fertilizer, we want to make sure we tell them which fertilizer, how much of each of the liquids are we giving it, the water, the Coke, the L8, and the Mountain Dew. We want to maintain all those constants as best we can. What units that we're using, we want to be consistent with that. So anything that has to be consistent needs to be discussed. Okay, so consistency is important. And you want to talk about those because you might be aware of what you need to do, but whoever is performing this experiment may not have an idea about some of these things. So you want to make sure that you indicate those in your procedure. That has to be a part of your procedure, okay, how you're being consistent with that. And I can't overemphasize how important these details are. That shouldn't have popped up there. All right, so that's the front page. I'll come back with a second video about the results and the conclusion. All right.